So it is my pleasure to introduce our next speaker, Dr. Sebastian Marquardt. So Dr. Marquardt is quite the world citizen. So he's from Germany and did his undergrad there, but then moved to the UK for a PhD in the lab of Caroline Dean, working on lincRNAs in Arabidopsis flowering. He then moved to Boston for a postdoc in the lab of Steve Bertowski, who I've seen in the audience today. And there he switched over to working in yeast and studying there how chromatin limits divergent transcription. He stayed in the Boston area for a while as a visiting scientist in Mary Gering's lab, before moving to Denmark to start his own lab there at the Copenhagen Plant Science Center. So he's now had his lab there for about 5 years, and he's done important work studying transcription in Arabidopsis. And for this, he's been awarded an ERC starting grant, different funding sources from the Novo Nordisk Foundation. And most recently, he's also been recognized by EMBO for his outstanding work since he was selected as an EMBO Young Investigator. So with that, I am very excited to hear what science he has to share with us today.
Okay, well, thank you very much for putting this wonderful community together. So I would like to begin by acknowledging these wonderful people that I had in my lab, that are currently in my lab and used to be in my lab, that perform most of these experiments that I will talk about today. And along the way, we were fortunate to have wonderful collaborators that were around locally or internationally. And I'm very grateful for support from various funding agencies that supported our research. I'd like to begin by thinking about the amazing features of DNA in our cells. So for example, if you would stretch out the DNA that's in the nucleus of a human cell, it would be about 2 meters long, but somehow it manages to fit into the nucleus that has a dimension of a few micrometers. And perhaps a more relatable scale would be if you would think about the same problem, to squeeze a 40-kilometer or 25-mile-long wire into the size of a tennis ball. So I think this illustrates what an amazing packaging job is going on with our DNA in our cells. But we also know that just packaging it very tightly is not enough. It also needs to be accessed in different cells and different tissues or at different types when we experience different environments. So there's an extensive need for dynamic regulation. And one important feature that we know about these chromatin compactions are nucleosomes. And yes, I know that in your series you have had many talks about nucleosomes. So just in brief, we know that the basic structure of nucleosomes are histone proteins that form octamers and wrap the DNA around them. And then these structures can then form higher order structures that really achieve this very efficient packaging. And what I would like to highlight are these sort of tails that I've drawn here, because these are N-terminal histone tails. And we know that these can be subject to post-translational protein modification. And that is interesting because a lot of this happens at, for example, at lysine residues highlighted in gray. And we know the most famous modifications are different types of methylation or acetylation. And one interesting aspect of this that these modifications provide a binding platform or a signaling platform for processes that act on the DNA, for example, regulate accessibility or other activities. And since these effects regulate the genomic blueprint of life, our DNA sequence, but act above the sequence, we also talk about epigenetic effects, and that can affect all aspects of DNA biology, replication, and so on. But I, in my presentation, will focus on RNA polymerase II transcription, and I will focus on chromatin signaling that goes on during RNA polymerase II transcription that happens in active, open chromatin regions. And I would like to show by sharing one cool data slide that was generated in my lab very recently. So we downloaded all, as you can see here on the right, all types of ChIP-seq data for Arabidopsis for chromatin marks that we knew were associated with active gene expression. And as you can see, it's very interesting to look at this because you can see this profile of histone modifications along a transcribed gene. And this is one of those meta-gene plots where you try to understand the entire genome and condense all the information on one representative gene. So it's in many ways an oversimplification, but it's nevertheless useful. And when we go through this from left to right, you can see that here is where we sort of would initiate transcription. This is where we have the activity of many sequence-specific transcription factors that come in to recruit the polymerase. And then here in this area, we also find this nucleosome depleted region that characterizes promoters, where if you look at these marks, there's very little on since there are hardly any nucleosomes. And then when we move into this green zone, it gets more interesting. That's where we encounter sort of the first well-positioned or +1 nucleosome. And in this sort of zone is where we can find very, very many of these marks peaking. For example, we find here, this is essentially all histone acetylation marks we looked at, they seem to be here, or for example, also HTK4 trimethylation. And then when we move into this purple zone, I've shown that now we bring in some of these factors that are needed for RNA polymerase II transcription of chromatinized templates. And I would like to highlight, for example, this FACT complex, which is an important histone chaperone that helps to move the polymerase to transcribe. And you can see that the chromatin environment during transcription in this zone is very different from here. And I mean, there are some very interesting differences for the aficionados that we can maybe take up in the Q&A session. But I would just like to highlight that for plants, a very good marker for elongation is HTK4 monomethylation. And if you're trained with the research in other systems, you would, for example, expect to find HTK36 trimethylation here. But this mark in plants is actually at a different position where you would expect it. It's sort of more at the beginning. And when you are then coming towards the end, you come into another nucleosome depleted region where we recruit a lot of 3' information and termination factors. And so this chromatin signaling that goes on during Pol2 transcription is very important because it helps the polymerase to discriminate where along a gene it is. And this is then linked to the recruitment of pre-mRNA maturation factors. For example, it helps for capping to take place at the beginning, termination at the end. And one other important aspect is chromatin signaling that represses initiation from within coding regions. And one thing I'd like to note is, for example, the low histone acetylation levels in coding regions. Okay, so and I would like to sort of say that you don't have to remember all the details of this because I have introduced this color code here that maybe helps to just think about the system when I'll show this color code. And I'll now tell you about the sort of significance of this positional information system based on chromatin signaling. And it is one important model how we can interpret functions of non-coding DNA. And we got into this when we studied the role of a link RNA in budding yeast, CAT60. And so when we have CAT60 mutant, you can see that yeast grows very poorly, particularly on non-fermentable carbon sources such as glycerol. And this indicates mitochondrial defects. And there's a very strong link to mitochondria because the ATP16 gene functions as ATP synthase in the ATP synthase complex in the mitochondrial membrane. And we knew that certain mutants in ATP16 can lose the mitochondrial genome. So we also checked whether the CAT60 mutant have lost the mitochondrial genome, and this was indeed the case. So we have a mutation in the nuclear long non-coding RNA that loses the mitochondrial genome. And then we thought this could be linked to ATP16 expression, and so we look in wild type on a northern blot shown here, and you can see that you can detect the gene expression, but when you look in this mutant, it is gone. And then we also looked at certain RNA degradation pathway mutants like XIN1 shown here. And this was interesting because in this mutant, we could see that there was still some residual transcriptional activity of this locus, but we could detect an 5' extended read-through transcript. And to understand what was going on, we thought initially of two main models. So this could be a function of the CAT60 non-coding RNA, or perhaps the fact that there is a non-coding RNA is a big distraction. And what's really important here are cis elements in the DNA. And so both of these models can be tested quite easily. And when we got the results, it actually showed that this is not the answer. So instead, we thought of additional ideas, and we thought it could be possible that what's important here is the efficiency of CAT60 termination. And so we thought of experiment how we could increase termination efficiency when we had these read-through transcripts and tested if this could induce ATP16 expression. And we also tried to think of other strategies to have efficient read-through transcription. And cut it short, both of these strategies worked. So I would like to summarize it like this. So when CAT60 terminates efficiently, the sort of co-transcriptional positional signal can establish over this gene and promote gene expression. But when termination efficiency is low, then these promoter DNA sequences that are the same here and here, they now become under the influence of this chromatin signaling during transcriptional elongation, and just represses functional expression of this ATP16 gene. So essentially we have uncovered connection between non-coding transcription and genomic punctuation. And if you like, you can extend these analogies to punctuation and language, just illustrated by these two sentences here. So if non-coding transcription terminates efficiently upstream, you carry the promoter instruction to initiate. But for example, when the environment changes and non-coding transcription is inefficient, you have read-through transcription over the downstream gene promoter. And this now overrides the DNA sequence information that is still there with chromatin signaling associated to elongation and represses functional gene expression. So I would like to say that this type of gene regulation is very well studied in budding yeast. And so we have, for example, recently wrote a review where you can look at the open access link here, where we discuss many of these examples. But there's also excellent other literature, and I would like to give what literature that I've personally enjoyed very much is, for example, from the Fred Winston lab. So if you have the opportunity, I would recommend you read some of the papers if you're interested in this mechanism. Nevertheless, I would also like to point out that even though this is a very important mechanism in yeast, I would make the argument that it is understudied in more complex eukaryotic systems. And we like to understand more about this in plants for a lot of reasons, but one reason is that it's very clear that if you're working with plants, you are working with a system that is very good at environmental interactions. Of course, many organisms can just move away from environments they don't like, but plants, they have to be very good at dealing with everything that's thrown at them and find some way to do regulation to come out on top. And so in the second half of my talk, I'll tell you what we learned about this chromatin signaling during elongation in plants. And so the first thing we were doing, we were looking at some insertional mutagens that can integrate in the plant genome and sometimes drive expression into the rest of the genome. And then again, you can have these cases where you have transcription into the downstream gene, and this results in a very interesting type of gene repression. Because by many mechanisms, or by many assays, I should say, it would look like this downstream gene is actually overexpressed. But in fact, it is a recessive loss of function mutant. And that has to do with the fact that, again, you target these chromatin signaling associated with elongation that repress initiation from this promoter. And in our case, we like to work with this allele of this Quasimodo mutant, where you can see it has a very nice visual phenotype here, that it's some staining phenotype because cell adhesion is defect. And that allows us to do some nice genetic work where we can use various approaches to introduce mutants in this system and look for plants that look more wild type again. And so the first mutant that we find are mutations in this FACT complex that we knew from work in budding yeast and other work is part of this sort of PO2 elongation complex that also is involved in this type of repression. But one interesting aspect of this was that when we had these mutants, so we could restore the downstream gene expression, but we could see that the overriding chromatin information was still there. So there are different types of how the suppression could work. But this was actually an interesting one because somehow this indicated to us that FACT was somewhere in between the signaling and repression. And so we were then wondering how much of these the effect of FACT we could uncover when we studied this in the endogenous gene context. And for this, we worked with Vicente Pelecano, and we performed transcript isoform sequencing in the Arabidopsis genome. And so transcript isoform sequencing is a short read-based method that you can use, and you can identify the beginning and the end, so the 5' and the 3' end of one RNA molecule. So it allows you to identify gene isoforms on the basis of these two ends. And when you look at this, you can see that a lot of this variation in plant transcript isoform diversity is indeed explained by transcripts that start in the middle of genes, and then they go all the way until the end. So this indicated to us that it would be worth investigating if FACT and this elongation signaling plays a big role here. And here I'm showing you some of the data that we obtained. So I should say that this is just a screenshot of an example gene where we have a promoter that is sort of at the endogenous that represents the promoter TSS that is usually strong. And then we found 17,000, so I would say quite a high number of transcription start sites in the middle of genes, as shown here in red, where you can see that they're upregulated in various types of mutations that disrupt the function of the FACT complex. And I will also say, so this is transcription start site sequencing data. So it's a quantitative method that focuses on transcription start sites. And you can then also look at TIF-seq data. This is this method that shows you these connections. And here you can see that the start sites that get used in these FACT mutants, that really they come up in these mutants and then they go all the way until the end. So indicating that many of these isoforms in plants are repressed by FACT-dependent chromatin signaling. And we then tried to learn a bit more about the specification of these transcription start sites and the mechanism of repression. And we performed several of these sort of plots, which are again sort of some metagene plots where we look at the center of the transcription start sites and compare those that happen at gene promoters to intragenic TSS that we thought are independent of FACT and FACT-specific regulation. And as you can see here, so when we found for HEK4 monomethylation, of course, that is very low HEK4 monomethylation in plants promoter regions, but we found a small but significant increase at FACT-specific TSS with this HEK4 monomethylation signature. And so we think HEK4 monomethylation works together with FACT in the repression. But when you look at this effect, you also can have to admit that it's not super huge. So we are very sure that this is not the only mechanism of specification, and it likely works with others that we are currently trying to identify. And so to summarize this part, so we've worked with these systems where we can study the repressive effect of RNA polymerase II elongation on transcription start sites in gene promoters. And we think this includes the activity, this requires the activity of FACT linked to HEK4 monomethylation signaling. But our genome-wide analysis also suggests that this is not a mechanism here that's specific for these read-through contexts. In fact, we find a lot of examples, thousands of examples, where this type of regulation, chromatin-based regulation, is used to determine what gene isoform is made from a single gene. So it's a method to diversify the sort of RNA information that you can derive from your DNA information. Okay, and so now I'd like to, in the last part of my talk, I'd like to talk about sort of genomic punctuation effects at gene boundaries. And we got into this when we developed a method to map RNA polymerase II transcription by mapping the last 3' base that got added to the polymerase complex. So that's similar to a method that I had used before in yeast, and we call it plaNET-seq in analogy to this method. And I would like to show you here, this is sort of an example screenshot for a plant gene based on this plaNET-seq data. And you can see here we use a mock IP control that helps us to believe in the specificity of our signal. And you can also compare this to RNA-seq reads shown below. And I'd like to highlight 4 points that we can see here. So the first thing that you can see is that you see this big accumulation of reads very close to the transcription start site. So this is how it looks like when you look at something like promoter-proximal stalling for a plant gene. But this is not the only time we see in sort of accumulation peak, and then there are some valleys. So this goes on throughout the entire gene length, and these could all be potential sites of regulation for POL2 progression that we could not really look at before. So now we have the opportunity to consider whether this could be some type of regulation that is going on. And another important aspect is that you can study POL2 transcription beyond the poly-A site, for example, that you would see here as the end of the RNA-seq reads. So we have the poly-A site, but with this method, we can pick up this transcription beyond the poly-A site. And that is important because we know that this type of transcription is linked to the termination process and could extend to trigger these sort of gene regulation effects that I've been talking about. And the fourth aspect I would like to highlight are these red reads. So with this method, we are also able to pick up various types of non-coding gene isoforms that are generated, for example, if they're on the antisense strand. Okay, to talk a bit more about this pausing or stalling part. So we found that when we anchored the nascent transcription sequencing signal at various features, we found a very strong correlation with the center of the +1 nucleosome. So we believe that plant promoter-proximal stalling is nucleosome defined. And that is interesting if you're familiar with the metazoan literature, because in the metazoans it's believed that you have 2 stalling or pausing peaks. So the predominant pausing peak is actually further closer to the TSS and specified by a metazoan-specific pausing complex. But that pausing complex is absent in plant genomes, and you still detect this sort of stalling signature, but it's nucleosome defined. But that made us curious what could be sort of going on here, these stalling regions during this gene expression. And what helped us to learn about this was when we looked at transcript isoform sequencing data, TIF-seq data, in nuclear exosome mutants. So this is the same complex that many people use as a trick in yeast or mammalian cells, but we can also look at some plants. And here's a screenshot of a plant gene where you can see in wild type it has one main gene isoform. But when you look at this nuclear exosome mutant, you can see this population of short transcript that start at the same position, but then terminate very quickly after initiation. And so we find that these transcripts are about 90 nucleotides on average long, and we call them short promoter-proximal RNA or SPP RNAs. And they are very abundant, so you can also pick them out in these sort of length distribution plots when you just look at these read distributions in this mutant. And so we thought there seemed to be a spatial coincidence with these SPP RNA formation and promoter-proximal stalling. So then we look at this more carefully, and here we look at this nascent transcription data anchored at the 3' end of these SPP RNAs. And as you can see, you generate these SPP RNAs really very beautifully just after this peak of stalled polymerases. And so we began to consider that it could actually be a real connection between stalling and termination near transcription start sites. And if that was true, we should be able to identify differences between genes for which we can find SPP RNAs and genes that didn't show them. And so for that, we compared nascent transcript sequencing data for genes with SPP RNAs in blue to expression match control genes here in orange. So these two cohorts, they have the same overall nascent transcription profiles, but it's just that the blue genes have SPP RNAs. And you can see that the blue genes that have SPP RNAs, they have much more pronounced promoter-proximal stalling. So there seemed to be a very good correlation between stalling and SPP RNA formation close to transcription initiation sites. And so to summarize this a little bit, so we developed this nascent transcript sequencing method, and this helped us to define and identify this nucleosome-defined stalling in plant genomes. And we could also show that this coincides with the formation of a new non-coding RNA species, SPP RNA formation. And it's interesting to think about this because the other main stalling peak that we know of in genomes, not only plants, is actually at the 3' end, and we know about the function there, because there it helps the polymerase to terminate transcription. So our data suggests here that there seems to actually be these two stalling peaks. They may have a unified purpose, that is to initiate transcriptional termination. So, but I should also mention that we are still in the early stages of trying to understand what these SPP RNAs are trying to tell us, and there are many different models that we are pursuing. But it seems to be that SPP RNAs are positively correlated with gene expression. And when we try to go in and mutate some of these SPP RNAs, the gene expression of the mRNA actually decreases. So it's not like a negative correlation as you might think. And one important aspect of when you think about stalling or pausing is always that it sort of comes along the idea that there has to be at least a locally different transcription speed. We were interested in testing this by having a mutant that is not able to slow down. And that's actually something that I initiated during my postdoc. So we started to collaborate with Craig Kaplan, who's an expert in mutants in the polymerase trigger loop that alter the transcription speed. And so 8 years ago, so it must have been, or maybe 9, it must have been 2011, he shared like a point mutation in the second largest subunit of the yeast polymerase with us here that he thought would accelerate transcription speed. And as you can see here, like 9 years later, we were able to publish this. And so we were able to integrate this accelerating mutation into plants. And then we looked at nascent transcript sequencing profiles. And here's sort of some screenshots where you can see when you accelerate transcription in these mutants, you beautifully lose the stalling. And this is also true for like basically looking at also every gene, like on the metagene level. So you have a very strong effect on the stalling peak that you reduce, and instead you run much more into the gene. And this effect was not specific to the promoter-proximal stalling. In fact, you also see this for stalling at the 3' end. that is very strongly reduced here. Again, you can see this very beautifully on these metagene plots. And instead, you see for the aficionados that look at this more, it looks very clear that you have much more read-through transcription. So when you accelerate transcription at these gene boundaries, you lose both stalling peaks, and this results in read-through transcription into the gene body and beyond. But one aspect that was particularly interesting for us is that we were able to actually work with these entire organisms. So when we work with plants, we can try to let them grow, even though they have some of these very drastic mutations. And here's how this looks like. This would be a wild-type plant, and this would be a plant that only have like fast polymerase. And when we looked a bit more deeper into these molecular features, we realized that this mutation is very likely resembling an autoimmune phenotype. And this, we could see this based on various markers, and there seems to be a very, very nice finding. And if you put this into a bigger context, this is interesting because we know from many human viral infections, they seem to directly target 3' information complex, which could again then lead in this massive read-through transcription. And we also know from plants, one important antiviral defense mechanism is the RNAi pathway that can also in part be triggered by effective 3' information. So there seems to be a very interesting connection between the organismal stalling at gene boundaries and the immunity. Yeah, so to summarize this a little bit, so our sort of these fast transcription mutants was very interesting for us because it linked sort of these main two stalling sites, the gene boundaries, to autoimmunity. And of course, in these type of mutants, you have a lot of read-through transcription, and it could of course affect gene expression to these mechanisms I've talked about by overriding the promoter chromatin signature and affect gene expression. So overall, I've introduced you to this sort of chromatin signaling during elongation in plants, and there are some interesting differences also to other eukaryotes. And this is an important model to interpret how non-coding DNA can function, because sometimes it could just be the act of non-coding DNA transcription that is important, and that has important implications when you're interested in, you know, sequence conservation versus actually the sequence here for these mechanisms does not matter as long as there is a positionally conserved non-coding transcription event. And when we studied this in more detail, we found that this chromatin-based regulation that ties into this gene repression by read-through transcription is very important to regulate gene isoform diversity. So the same chromatin players that we could identify have profound effect on which gene isoform is made. And of course, this could be diversifying the proteome by generating gene isoforms that lack important N-terminal signaling peptides, for example. And then when we looked at the gene boundaries, we could resolve very nice promoter-proximal stalling and stalling at the 3' ends that in both cases seem to be linked to transcriptional termination. In one case of these short promoter-proximal RNAs, but in both cases, there seemed to be the need of the ability of the polymerase to slow down, and otherwise you would trigger this autoimmune phenotype. And with that, I would like to thank you for your attention.
Thank you, Sebastian. That was fantastic. Okay, I have so many questions already, but I'll start. Like, there's questions from the audience, and in the meantime, like, if people want to raise their hands, they can do that. Just as a reminder. But first, there's a question from Peter Sarkis, and he's asking, do you have any idea on the stoichiometry of A3K4 monomethylation? So the increase you see, like, between the genes or between the groups is pretty small. And he says, like, much less than 1 H3K4 monomethyl per nucleosome. Could it be a subset of genes showing a larger difference, or are all genes affected in some of the cases, or like in part of?
Yeah, no, that's a very good point. I mean, I think that we don't. We have looked at this for some example, but I mean, it seems to be for the example that we could study in a bit more detail when we went in with additional techniques. It did not seem to be like an amazingly huge difference. So I think it is. I don't believe that this cannot be enough for specification. So there has to be something else. But what exactly that is, and also how these differences are mediated between these two types of transcription initiation sites within coding genes, we don't really know yet. That's something we're trying to identify. But for us, it's already progress that we're thinking about H3K4 monomethylation, you know, over other types of signaling that we might have expected if we are only informed on other systems.
Then there's a question from Chandan Kumar, and he says, "Great talk, Sebastian. I have a question about the overall expression of RNAs of the fast mutants. Mm-hmm. So if the RNA pull-to stalling is not happening, is then RNA expression overall increased?"
Yeah, so that's I mean that's a very interesting question, but it's not so easy to answer. I mean, we've performed some RNA-seq and so on. That's also part of this publication. I mean, there, of course, you find a lot of gene expression differences, but I mean, most genes seem to be. I mean, there seem to be a lot of common. The gene expression program did not seem to be like completely going crazy. So it seemed to be most genes seem to be expressed as they are in the wild type. I mean, we focused on some differences in splicing. That's also the idea that the transcription speed affects splicing. So we looked at this in more detail. But yeah, surprisingly, it looked very similar, I would say.
I was wondering myself, when you saw the read-through transcription with plaNET-seq with the faster mutant, do you know whether cleavage is still happening? So like, is it that it's not getting cleaved and so the polymerase is never chased off, or it's still getting cleaved but the polymerase can get further, in the torpedo model, like it would take longer to be chased down, for example?
I mean, we interpreted it as if the torpedo model takes longer, but I mean, technically, I think we have not with this. Well, I guess we would maybe also see it on the RNA-seq. So I don't think we see a big difference in the 3' and in the RNA-seq. Okay. So I would interpret that as sort of the torpedo situation. Yeah. But that would be my answer there. But I mean, I think we haven't looked very hard. But we've looked at the average sort of read-through lengths downstream of the poly-A site, and so the average gene sort of has 500 nucleotides downstream, and in this fast mutant, it is much longer. I think it was like 150 nucleotides longer or something like that. So that's an analysis we did.
Okay, so I think it's already past the hour, so we'll leave it at this for the Q&A.